Hello, this is Jonathan Schwartz. I'm the Chief Medical Officer at Rocket Pharma, and over the next few minutes, I'll provide an update with respect to the results of the ongoing gene therapy clinical trials that are taking place in the US, Spain, and United Kingdom. These are results that were presented recently at the American Society of Hematology meeting in December 2021, with some additional updates from January 22. My co-authors from Stanford University, University of Minnesota, Hospital Infantil Niño Jesus in Madrid, and Great Ormond Street Hospital, University College of London, as well as CMAT, also in Madrid, are listed here on the slide. Before providing an update of the actual clinical trial results, let me place the studies in a little bit of context. The original clinical trial, the Fanconi 1 study, was conducted exclusively in Madrid, Spain, as an investigator-initiated trial. The current studies, sponsored by Rocket Pharma, include 3 trials that were developed to be companions to one another, and this includes the European Fanconi 2 study and the United States-based phase 1 and phase 2 clinical trials. The essential question underlying each of these coordinated clinical studies is as follows. Is it possible to prevent the development of bone marrow failure in Fanconi complementation group A patients whose hematopoietic stem cells are collected and reinfused early in the course of bone marrow failure? The diagram on this slide is a cartoon illustrating the progression of blood counts and bone marrow status in the majority of Fanconi patients during the early years of life. In the majority of Fanconi patients, blood counts will decrease during the initial decade of life, and the same is true with respect to the number of hematopoietic stem cells in the bone marrow that are responsible for making blood and bone marrow cells. In general, an allogeneic transplant is performed only when a patient has entered a relatively severe phase of bone marrow failure. Frequently, this is done around age 8 years of age, although obviously everybody is different. And the transplant is enabling a very rapid correction of the blood and bone marrow status, usually identified in the months immediately after the procedure. Gene therapy is a bit different, as it is intended as more of an early stage preventative intervention, typically done when there still are a reasonable number of blood stem cells that are available to enable a genetic correction. The intent of gene therapy is to correct blood and bone marrow deficits without conditioning, and potentially to do so requiring very limited hospitalization or transplant level medical care. The hope is that gene therapy would be able to enable this correction without any additional increase in subsequent malignancies like head and neck cancer. And hence, this is really intended as an early stage preventative measure. It's not necessarily at present a substitute for an allogeneic transplant, which is one of the critical therapies of choice once somebody has encountered a severe stage of bone marrow failure. The gene therapy itself involves insertion of a functional correct copy of the FANCA gene into a patient's own bone marrow stem cells, which are also referred to as autologous CD34 cells. And this is intended to enable these cells to then be resistant to DNA damage and to repopulate the bone marrow and blood because of a documented proliferative advantage of gene-corrected Fanconi cells over their uncorrected counterparts over time. And this this process of gene insertion is known as transduction. The cells are infused into a patient, and when they take root in the bone marrow and begin to produce bone marrow and blood cells, that's a process that's known as engraftment. The actual therapeutic procedure is illustrated in the cartoon on the right side of the slide, and this involves a series of steps over a period of 1 to 2 weeks. The first step is mobilization of bone marrow stem cells or CD34 cells using 2 medicines, GCSF and plerixafor, which are given over approximately 6 days, and collection of stem cells from the peripheral blood over 2 days using a procedure that's known as apheresis, which is kind of like a large-scale transfusion type of endeavor. It takes several hours and is done over 2 days consecutively. Once the cells are collected from a patient, the CD34 cells are further purified to enrich for the cells most likely to be long-term stem cells. Those stem cells are transduced in a specialized cell processing laboratory with the therapeutic vector containing a correct copy of the FANCA gene. And then after several days of this, really 2 to 3 days, those cells are then sent back to the hospital and infused into the patient through an intravenous catheter. And then the cells themselves are responsible for finding their way to the bone marrow and then taking root. And all of this occurs without any chemotherapy conditioning. This slide illustrates some of the key eligibility criteria and endpoints for the 3 clinical studies that are underway. This is a study that's for patients with complementation group A who are at least 1 year of age. Additionally, patients are not entered into this study if they have an available and eligible HLA identical sibling donor, in other words, a sibling matched donor. Also, other exclusion criteria are patients who have developed myelodysplastic syndrome or leukemia, or patients who have evidence of mosaicism in the bone marrow with stable or improved blood counts. There are several efficacy endpoints, which is another way of saying ways that we are attempting to measure whether the treatment is successful. One of these endpoints is phenotypic correction. In other words, do the bone marrow and blood cells now behave as though they are resistant to DNA damaging agents? And in fact, the resistance of bone marrow cells to mitomycin C, which is one of the DNA damaging agents, is the primary endpoint of the study. Other very important endpoints include evidence of engraftment. And we measure that in the blood and the bone marrow by looking at something called vector copy number, which tells us whether the cells in the bone marrow and the blood actually have copies of the correct FANCA gene. We expect and we hope that both of these measurements are ones that will increase over time. We also are looking at clinical or hematologic response, which is measured as preventing the development of severe bone marrow failure. And this is measured by looking at blood counts over time with the intent of stabilizing or even increasing peripheral blood counts in the years subsequent to therapy. Of course, always in conjunction with these efficacy endpoints, we're very closely evaluating the safety of the gene therapy itself, both in the hours and days following the procedure, but over the long term as well. As of February 2022, 11 patients have received gene therapy on the 3 Rocket-sponsored gene therapy clinical trials. 3 of these patients have fewer than 1 year of follow-up. And although we'll present some information about the age and location of the patients and the amount of cells that they received, it's too soon to really review information about blood counts or other efficacy parameters. For that, we'll focus on the 8 patients who have at least 12 months of follow-up. Those patients are identified by their unique number here. And very important to emphasize that of these 8 patients, one of the patients, this was a patient who was enrolled on the phase 1 part of the trial in the United States, was withdrawn from the study at around 18 months after therapy due to progressive bone marrow failure that required an allogeneic transplant. This was a patient who had developed a pretty severe influenza B infection around 9 months after gene therapy, and continued to have progressive decreases in her counts, requiring the transplant, which occurred without substantive complications, very fortunately. So this was a patient who did not have a therapeutic benefit from the gene therapy. We will in detail review the information from the other 7 patients who are being followed on the trial, and more information on those will be provided momentarily. This is a complex table describing a number of important features with respect to the patients enrolled on the clinical trials to date. You can appreciate that patients have come from the United States and Spain, as well as the United Kingdom, and the patients who've been enrolled have ranged in age from age 2 to age 6, with a pretty equal distribution of boys and girls. 8 of these patients have at least 12 months of follow-up, and we'll review more detail on those patients momentarily. Some of the other things that are listed in this slide are some of the drug product characteristics that we use to measure the cells that are being infused into a patient. One of those metrics is the number of CD34 cells per kilogram of a patient's body weight. And in general, we are striving to have at least 200,000 or 2 times 10 to the 5 CD34 cells per kilogram infused. And whenever possible, we're also looking for higher numbers, including numbers above 1 million, or 1 times 10 to the 6 CD34 cells per kilogram. We also look at the vector copy number in the cells that are infused, and in general are hoping for vector copy numbers that are close to or above 1, indicating that in the majority of cells, there's at least 1 corrected copy of the therapeutic correct FANCA gene. By and large, we were very pleased with the numbers of cells that we were able to collect and to infuse, and our ability to transduce these cells at high efficiency. And a great many of the cells in the cell product, 49% as a mean, were resistant to 10 nanomolar mitomycin C, indicating that they can withstand a reasonable amount of DNA damage. This is a very important slide. The left panel is a measurement of the vector copy number in the blood cells, the leukocytes or white cells, of the 7 of the patients enrolled on the clinical trial. And what we're able to state is that for this group of patients with at least 1 year of follow-up, some sustained increase in peripheral blood vector copy number has been seen in 6 of those 8 patients, which means that there's a percentage of cells in these patients' blood that have a correct copy of the FANCA gene. And in the majority of these patients, the trajectory of the percentage of these cells that have the correct gene appears to be increasing over time. The table on the right portion of the slide illustrates evidence of correction in the bone marrow and the level of cells from the bone marrow that were resistant to mitomycin C. At baseline, prior to treatment, all of the patients had 0 or close to 0 mitomycin C resistance in their bone marrow cells. And one can appreciate that for the majority of the 7 patients that are listed here, at anywhere from 12 to 24 months after gene therapy, there's anywhere from 0 in one case to 16 to as high as 63% bone marrow cells that are resistant to mitomycin C. So basically, we can state that peripheral blood genetic correction was seen in 6 of the 8 patients who have been followed for at least 12 months, and that concomitant bone marrow mitomycin resistance above 10% of baseline values, to 10% above baseline values, was also seen in 6 of 8 patients followed for 12 months or more. This slide illustrates the peripheral blood counts of the patients, of 6 of these patients. Each of the patients' blood counts are presented together. One can appreciate the little triangle is when the gene therapy was given. Everything to the left was prior to gene therapy, and everything to the right is after gene therapy. The overall white blood count is always illustrated in the upper left in purple. The hemoglobin is illustrated in red in the lower left. The neutrophils, which is a subset of white blood cells, are illustrated in the upper right. and the platelets in green are illustrated in the lower right. And by and large, in 5 of the 6 patients depicted here, we see stabilization of all of the blood counts, the white cells, the neutrophils, the hemoglobin, and the platelets during the period of time after gene therapy. Although in many cases, this is still early, and we want to be able to watch this for more than 1 year. In this subject 1001, there did appear to be decreases in neutrophils and some decreases of hemoglobin and in platelets during the initial 12 to 18 months after gene therapy, with some potential indication of stabilization starting at around 21 months for platelets, for hemoglobin, and also for the neutrophils. All of these patients have been well at home and without any need for blood transfusions. So our conclusion is, of the 12 patients treated to date, 6 of the 8 patients who have at least 1 year of follow-up display evidence of engraftment. 1 patient had a course that was complicated by an influenza infection and had progressive bone marrow failure, requiring a bone marrow transplant. Fortunately, this patient had a successful bone marrow transplant, demonstrating that one can actually receive a transplant if the gene therapy is unsuccessful. Increasing bone marrow mitomycin resistance has been seen in 6 of the 8 evaluable patients, and that has been accompanied by blood count stabilization in at least 5 and possibly 6 of these patients. Very importantly, the safety of the therapy appears favorable. Patients are treated without conditioning, and we have not seen evidence of any dysplasia or other concerning features. The one serious adverse event that was encountered was in 1 patient that was related to RPL102, was 1 infusion-related reaction, which occurred in 1 patient at a level of grade 2. Adverse events are graded anywhere between grade 1, the least severe, and grade 4, the most severe. This was an allergic reaction, which was transient and responded very rapidly to antihistamines and other Tylenol or paracetamol type medication. And by and large, a very, very uneventful safety profile. Very importantly, we have set the bar for this trial that efficacy in 5 of 12 of the treated subjects observed over the course of 1 to 3 years after therapy would be the benchmark by which we would consider the therapy as a successful preventative measure. Very importantly, these trials, these interventional or prospective trials, are also taking place in conjunction with several natural history efforts, including very detailed reviews of Fanconi anemia mosaicism that include 2 publications that have occurred over the past 2 years, indicating by and large that for Fanconi mosaic patients who have evidence of mosaicism across all the different blood lineages, prognosis of those mosaic patients appears to be very favorable. And that's important because in the gene therapy trials, we are attempting to essentially replicate this mosaicism or naturally occurring gene therapy. Another effort that's also underway and that will result in a publication very likely in the early portion of 2022 is a comprehensive review via the IFAR registry that is looking at the general trajectory of blood counts in children with Fanconi anemia, complementation group A, over the initial years of life. And this will serve as an essential comparator to the blood counts that we see in the gene therapy trial. And this is a publication that is nearing submission from Agata Smogorjuska at Rockefeller University and her colleagues that will likely also provide a lot of very detailed insights into the course of blood counts in Fanconi patients in the first year of life and beyond. Many of you are probably also curious as to what's next. And we'll spend more time on that during the live question and answer session. Very importantly, the clinical trials that are underway will be expanded by 2 additional patients who will be treated in the United States, bringing the overall number to 14. It is likely that those 2 patients in the US will be able to be treated this year in 2022, and more details on that in the Q&A. Very importantly, all of the patients on the Rocket-sponsored studies and the Franklin 1 study in Spain will be participating in a long-term follow-up study enabling 15 years of follow-up after gene therapy to study both the long-term effectiveness and, very importantly, also the long-term safety of gene therapy. Other clinical trials and other and similar endeavors that could be activated once we're at a point in time where we're confident in the overall safety and effectiveness from the current studies could include prospective gene therapy trials in complementation groups C and G, as well as an expanded access program for Fanconi A, which would allow for therapy with the current program for patients who are not part of the clinical trial, but would enable the patients, interested and eligible patients, to receive gene therapy in the period before there were potential health authority approvals in the US and Europe. Additional questions that we're hopefully going to be able to answer would also include whether the addition of non-genotoxic conditioning, in other words, conditioning with antibodies that are not chemotherapy, could enable more rapid or more consistent engraftment of gene therapy, and additional studies looking at hematopoietic stem cell expansion, which could involve L-trombopag or other more novel compounds that could increase the number of hematopoietic stem cells either within a patient or outside of the patient to enable gene therapy to be applicable in patients with smaller numbers of stem cells who are in more advanced states of bone marrow failure. Again, we'll discuss more of these when feasible during the live Q&A session. For Rocket Pharma, Fanconi anemia is a really integral part of our DNA. This is my 6-year-old keyboard from my computer that I was given when we started the company back in the beginning of 2016. It's no accident that the 2 of the 3 keys that are completely worn out are the F and the A. We're trying to wipe out FA one keystroke at a time. I had to retire that computer because the battery was shot, but I promise I'll be pounding away at the F and the A on the new machine. And the same holds true for all of us at Rocket and all of our collaborators. And speaking of collaborators, it's a large and important group, which includes colleagues in Madrid, in Minnesota, in London, in Barcelona, and some of them are mostly in the laboratory. Some of them are in the clinic, obviously in a huge debt to FARF and also the Spanish and German and UK societies, and all of the patients and families and the entire FA community, which really is an inspiration to any scientist or medical researcher hoping to make an impact against a serious disorder such as FA. Thank you for everything that you are and everything that you do. And one last thing, this is the Light Up for Rare campaign. We started it a few years ago when we got the Empire State Building to light up for Rare Disease Day, which is coming up on February 28th or February 29th, depending on which year we're in. And the goal of Light Up for Rare is to help spread the word and raise awareness for people living with rare diseases. Because even though each of those rare diseases is rare, collectively, they're not rare. It's something that touches on a great many of us. And as a result of this initial effort, we're now seeing many more buildings and landmarks and monuments illuminated around the world. And we hope that we can all take part in one way, shape, or form as we light up for rare at the end of February this year and every year. Thank you so much for your attention, and I look forward to addressing additional efforts in our question and answer session and anytime by means of internet, web, email, etc. Thank you and have a great day.